It's June, and in this presentation, I'm going to briefly introduce my project for CS221. The topic for my project is microbiome strain selection for cheese production. So actually, microbiome strains play a really important role in the production of cheese. They break down the proteins in the milk into amino acids and then form new proteins using the amino acid building blocks. The following is the task that I want to solve in this project. Given the amino acid sequences of the proteins encoded by all the expressed genes of a bacterial strain, I want to predict the taste quality of cheese fermented with the strain. So let's say we have a microbiome strain. It could have a lot of expressed genes. Through sequencing techniques, we can obtain the amino acid sequences of the proteins encoded by all the expressed genes of the microbiome strain. So now, one of the first challenges is to engineer good features from these protein sequences. How do we get numbers from strings? The second challenge is to build a good predictor to predict the taste quality of the cheese product based on the numerical features. I would say personally the most challenging part of this project is actually the feature engineering part. How do you actually design a good feature extractor that operates on protein sequences? So here's the data that I'm working with. So I have a variety of strains. For each strain, we have taste score ratings for a few different flavors, such as sweet, sour, bitter, and overall. And we also have the protein sequences for each of the strains. Let's just say that we just consider all the strains that score the highest on the sweet flavor. Now, if we look at the protein sequences of these strains, and if we look at the common sort of sequence patterns of these strains, then it's reasonable to assume that these sequence patterns could be related to the generation of sweet flavor. Therefore, the following is the approach that I took for feature engineering. So I take all the proteins that are unique to strains that score highest in a particular flavor. I use Seqfire, which is an open source software for protein sequence clustering. Through clustering, I have a few different clusters of the protein strains that are based on the sequence similarities. So now, if we look at each cluster, what we can do now is we can look for common patterns among these strains. In this project, I employed an algorithm called multiple sequence alignment, or MSA. This is a dynamic programming algorithm which helps us identify blocks of similar patterns. So, for instance, let's consider these 5 positions. And so in the first position, there are lots of Q's and a V. In the second position, there are 3 T's and a K. In the third position, there are 3 S's and an R. In the fourth position, there are 3 Q's and a V. And in the fifth position, there are 3 T's and an N. So what we can do is we can build a template for this 5 amino acid subsequence pattern, where at each position, we give an estimate of the probability of each amino acid showing up in that position. So now, given a new protein, we can scan the sequence of this new protein to find subsequences with similar patterns. So that is the basic idea of the feature extraction process. Because we do not really have a lot of data in this project, I therefore decided to use some relatively simple predictors because we only have roughly 200, a little bit more than 200 strains. And so that might not be like nearly enough data for deep learning based approaches. So the few different I tried a few different machine learning algorithms for the prediction tasks. These include random forest, support vector machines, gradient boosting machines, two nearest neighbors, and an ensemble model of all the above models. What I found was that actually decision tree based algorithms such as random forest and gradient boosting machines work much better than the other algorithms. My hypothesis for why this is the case is that genes, they are known to interact with each other. So a lot of times it's not necessarily that a particular gene works to produce a particular effect. It's often more like a few different genes, a particular combination of genes, they work together to produce a certain effect. So with decision tree based predictors, because we do these like hierarchical splits, it's more easier for us to take into account the different combinations of the different genes. And therefore they can better reflect how the genes interact with each other, and therefore we can expect these decision tree based algorithms to perform better.